Thankful to be here today. Thank you to the organizers for giving me some time. So I'm going to talk about gene therapy today. I'm a member of the Scientific Advisory Board. I'm grateful to be able to share some of the things I've learned over the last decade working in drug development, specifically for rare diseases. And I've been intimately involved in leading an AAV9 gene therapy program for another neurodevelopmental disorder called CDKL5 deficiency disorder. It does share some similarities with FOXP1, also some differences. So I thought I would use the short time I have today to just really go through the basics. I'm going to go through it pretty quickly, but I want to try to boil it down so it's pretty simple to understand, hopefully. But if you do have questions, please do try to find me. I'll be here during the lunch hour. I'm happy to answer questions that you might have. So the outline today is I'll start by explaining what gene therapy is. We hear this a lot. I'll talk about the main gene therapy approaches. How do they each work? We'll talk about how do we engineer a recombinant vector, that's the actual drug that you would take. How do we deliver the gene therapy to the body? And then once you get it inside the body, how does it get inside of your cell and do what it's supposed to do? So gene therapy treats disease by transferring corrective genetic material to a patient. So simply, in order to understand that, you need to understand what is genetic material. And I'm sure most of you know this, but just so that we're all on the same page, basically all of our genetic material remains inside of our nucleus of the cell and in the form of a chromosome. So the chromosome is just one large continuous DNA molecule, all kind of scrunched up into that shape. If you unravel that, a gene is really just a segment of DNA. And really that's a code. And so you might be familiar with the A, T, C, G. Those are letters that provide a code to the cell to make a specific protein. And the protein is the thing that really does the job. That's the thing that's important. So the genetic material in gene therapy is typically delivered by modified viruses that cannot replicate or cause disease. And so that's really important. It was actually a really amazing discovery in that you can take a virus, such as adeno-associated virus or AAV, that's something that normally infects us, but it doesn't make us sick, it doesn't cause disease. And what researchers have done is take out the normal genetic material from that virus and put in a therapeutic gene. So in the case for what we all care about, that would be FOXP1. So you're really just using that virus as a way to get inside the cell as a carrier. And then that delivered genetic material will enable the cells to produce the functional protein. So at that point, you're really just taking advantage of the normal machinery in your cells that works just fine. It just needs the code. It has the wrong code for FOXP1. And so it's not making a protein that works. So we're just going to put in the right code and then let the body do the work. So there are a number of different sort of categories of gene therapies. And of course, there's also antisense oligonucleotides. And so I'm not going to talk about that today. I'm going to focus more on the viral gene therapies. But I know Sumit will talk later a little bit about that because that's something the foundation is funding as well. So gene addition is where we'll add back a functional or a wild type copy of a mutated or missing gene. So that would be the case for what we would want to do for FOXP1. It's not working, it's broken, so we're just gonna leave it. It's not doing anything bad in the cell, at least from what we understand right now. And so we'll just put a new copy in, leave the old one, and that will make a new functioning protein. That should fix things. In another example, in some other disease states, you've got a mutation that causes the protein to take on a new function. So not only does it not work the way it should, it's also doing something that it shouldn't. And in that case, you need to inhibit it. And so we have ways and sequences we can put in there to turn that off so it stops doing the bad things. And then we also have editing. And so in this case, we are editing a mutated gene back to a functional or a wild type copy. And that's a little bit different. So here, what you're used to hearing about is CRISPR-Cas9. And that is actually working on your host cell DNA. All of the first two examples are actually putting DNA, exogenous DNA, that we're putting back into the system. So how do we get this therapeutic genetic material into the cell? So I alluded to this a minute ago, using these viruses as carriers. You can think of them kind of like taxi cabs. So a vector really is the vehicle used to bring the outside genetic material into the cell. And there are both viral and non-viral delivery methods. So you may have heard of liposomes or maybe lipid nanoparticles. These are non-viral ways of getting the genetic material into the cell. The problem is that they're not very good at hitting the brain. So they're pretty good at delivering genetic material to the liver, and they're being used successfully that way now. But we still are tweaking the science and trying to figure out a way for it to actually carry things into the brain, which is what we care about. For viral delivery, we have at least two options: the retrovirus or lentiviruses, or the adeno-associated viruses, the AAVs that you hear about. So the main difference between these two is that, sorry about that, is that the lentiviruses are integrating. So what that means is when you put that piece of DNA back in, it actually goes into the chromosome and becomes part of the normal DNA that you have in the cell. That can be a good thing because it will stay there for a long time, but it can be a bad thing because you don't know where it will insert. It randomly inserts, and it might insert into the place where there was something else that you've now broken. So if it inserts in the middle of another gene that's really critical, now that gene doesn't work, but you put back the FOXP1. So you're kind of fixing and breaking. For AAV, these are non-integrating. So when you put that piece of DNA back in, it actually sits beside the chromosome. So it exists as something, we call it an episome. It's a little circular piece of DNA, and it just lives there in the cell. It doesn't disrupt anything. The problem with that is if you have a cycling cell, it will get diluted over time. Lucky for us, the brain does not divide. So neurons do not divide in the brain. So if we can target those with AAV, they won't get diluted out. So what does it look like, this piece of DNA that I'm talking about that's inside of the vector shell or our taxi? There are a couple different components. The therapeutic gene, in this case, would be the sequence that encodes FOXP1. But we also need an enhancer and a promoter. These are just sequences of DNA that tell the cell to make that protein. They're kind of like the green go traffic light. And then we also need a poly-A signal, and that's kind of like your stop, that tells it to stop. And so this whole piece gets incorporated into that shell of the vector, which is our taxi, and then into the body. So how do we get it into the body? There are two ways. Typically for AAV, we'll use in vivo delivery. And so in that case, you're delivering it into the body, whether that's through IV delivery, intravenous into your vein, or into the cerebrospinal fluid. And I'll talk about that in the next slide. Ex vivo is typically used for lentivirus. That's where you actually take cells out of your body, then you treat them with the vector, and then you put them back into the body. This doesn't really work so well when you're targeting the brain. So what we would be thinking about is more on the left-hand side of your screen, the in vivo delivery. And here you have a number of different options on how to target the brain. We can actually go through the bloodstream. There is a small percentage of AAVs that cross the blood-brain barrier and get into the brain, but it's a small amount, so it's not very efficient. If we go into the cerebrospinal fluid, there are a couple different ways to access it. You can use something called ICV, which is into the lateral ventricle. There's a space at the base of your brain called the cisterna magna. We heard about this yesterday. You can put the virus in there, or you can target the lower back, the lumbar intrathecal, which is a little bit like where you'd put an epidural. And you can also do intraparenchymal delivery, and that's where you actually inject the virus directly into the brain. This is really good if you have one little specific region you want to target, not if you want to target the whole entire brain. So once it gets in, as I just described, how does it get inside of the cell? So it actually travels through the ventricular system. Let's see if I can, shown in blue, and it diffuses out into the brain parenchyma. So this gray part is where all of your nerve cells or your neurons are. When it comes in contact with the neuron, it gets endocytosed, so the cell kind of gobbles it up. It moves into this nucleus, and at that point, you don't need the carrier anymore. So the body just gets rid of that. It leaves the DNA or that code inside, and then the cell makes the protein. So this all probably sounds a little bit like science fiction. And I get that, but it's working. And it's still early days for targeting the brain. But there are examples of targeting other parts of the body, and spinal muscular atrophy, or SMA, has had one successful therapy. So there are 7 approved adeno-associated virus products. There are 7 approved lentivirus, and there is 1 approved CRISPR product. So we're getting closer to having this be reality. So I'm going to wrap up now. The key takeaways really are that gene therapy has the potential for halting, treating, or curing. Technically, this could be a cure. We're not there yet, for sure. There are a lot of hurdles that we're still working through, mainly targeting all of the cells in the brain. But technically, you're putting back what's broken, so it could be a cure someday. Success is really critically dependent upon solid basic and clinical science knowledge. So that's why the foundation is investing in thinking about a gene therapy now, because it does take a while to make sure that we get this right and we understand the science and we take our time. Each gene therapy strategy has its pros and cons. And right now, you probably noticed on the last slide, it's pretty expensive. So this is still something that we're working on getting the cost down. It's very expensive to manufacture these vectors, but over the last 5 or 6 years that I've been working in this space, I've seen the costs starting to come down already. So we're headed in the right direction. And that's all I have. Thank you.